I'm Peter Borak, Field Marketing Lead for the West at Cytiva. We are joined today by Mark Fox. Mark, please tell us a little bit about yourself and introduce yourself to the audience.
Hi, my name is Mark Fox. I'm the Senior Director of Protein Sciences here at Ultragenics, and I oversee the production of research material for all the modalities here at Ultragenics.
How do you manage discovery in a pharma environment?
The projects dictate that in many respects. So I think the goal is to try and challenge dogma to make a protein. And the proteins are all themselves very, very unique in what we have to do. So you're constantly trying to come up with innovative ways to make them. So you're not limited per se in innovation. You're more limited in the timelines and how quickly you can get it done. And that's a big driver, is the timelines, and that allows you to then innovate and get things achieved.
Well, and that sort of leads us into the next question. How do you manage the separate motivators between a corporate environment and a discovery mindset?
Being within a research department, you do have the advantage that you're starting from the very, very beginning of a project. So you have to be building that kind of discovery approach and innovating as you move forward. So they actually go hand in hand where my department is situated. So they're not separate as they may be in other companies.
How do you balance regulatory risk with innovation? Is that something that is a concern as you're starting from the ground? As you mentioned, you're in a discovery environment.
That is a concern. I think, as you're working forward, you want to make sure as you go forward on a regular basis that you're not innovating a brand new technique, which then you have to validate for the FDA. However, you're usually innovating around classical basic principles that already exist, which then doesn't push you into any of those regulatory roles or regulatory issues you may have further down the line.
Can you expand on some of those basic principles that you keep in mind?
Basic principles can be just as simple as aeration in a process for when you're growing your cells. That could be as looking at novel ways to make cell culture media. It could be different ways of using your modalities in your chromatography, taking standard analytics and maybe using them in a purpose that originally they hadn't been thought for, but still ask the same questions. So it's really that's where you can kind of innovate in many respects. To many, they may be seen as iterative, but from a basic principles point of view, or a lot of dogma that is out there, they're usually held within a box, and you're trying to see how far can I actually push many of these analytics or media or chromatography methods.
What is innovating in protein science?
Innovation is taking ideas from multiple fields and trying to bring them together. Well, I've worked in multiple fields, and I think that's one of the things that I can provide as an advantage, is ideas that come from AAV modalities, antibody modalities, enzymes, and other such approaches. And from that, you get exposed to lots of ideas, and you don't necessarily get a lot of crosstalk in our industry. And so by moving those ideas around, that I think then leads to innovation.
So Mark, I'd like to talk to you a bit more now about how you manage teams and your management philosophy. What is your management philosophy when you're working with scientists?
To identify what their drivers are. I know that sounds really cliche, but we're all human beings and we all have things that excite us more than other things. So I think scientists are particularly passionate, and the goal is to try and align their passion of what they want to achieve with a company goal. And I think that's the first thing. So identifying what everybody within the group really wants to try and achieve and where they want to go, and figuring out a path forward for them.
And how would you say you go about putting a team together?
A team is a collaboration. It's a team of communication. So it's trying to find people that will freely communicate. We'll actually stop being individuals and work as a team. And that's actually a really hard one in scientists, because, as we're going through the system, that's not encouraged. We're all be an individual. Don't be a team. So that's a hard one. Those are traits that are hard to find. So it's trying to find people that are enthusiastic, realize that you can't be an island. It takes a team to really get anything going, whether you're in academia or industry, and finding those personalities and people that will put that ego aside, and want to work to actually achieve a common goal. And that hiring is harder than than anything else.
And so to get to that end, what are your favorite interview questions?
My favorite question that I ask with people that work on bacteria is, what is your shake speed on your incubator, and why did you choose that? And for me, it's interesting the responses you get. But the standard question is because that's what I was told. Now, if we're supposed to innovate and do things, why aren't you questioning that speed that you're shaking your bacteria at? And that's always something that interests me.
So how do you select external partnerships, and what makes you say no?
Through my experiences, I've found that there are three buckets that generally external partnerships fall into. They fall into a bucket where you have to spend a lot of time managing them, and you have to provide them with a lot of guidance. Then you have a second bucket, which is a collaboration where you're talking science, you're innovating together, you're making progress. Both sides feel that things are going well. And then you have the third bucket, which is kind of like a black box. You have a partnership and you give them the scope of the work, and then they move away and they go and do it, and then they come back. And the problem I have with that one is you don't know timelines, you don't know what's going on, you don't know what's failing, so you can't work with them, and then you can't report to your upper management how things are going. So one and three are not my favorites. I can tolerate three. It takes a lot of my time, which can be a little draining, but the number two is the best. Finding an external collaborator which wants to really focus on the science, really wants to achieve something, and there's strong communication going back and forth. So, and that's how I tend to try and choose collaborators, and the other two format, depending on the cost-benefit analysis, I try to avoid.
Do you have any sage advice for folks who are starting in the biotech business, and how they can give themselves the best opportunity for success?
I think challenge dogma. I think that's the best bit of advice that I was ever given. You know, we all assume the things that we read in the papers are correct. Challenge them, do the experiments, design very simple experiments, challenge them, and constantly be trying to move forward. And it just takes time and hard work, but you'll get there eventually.